Hello and welcome to this Nature Research custom media webcast titled Tour to HCV Neutralization Space. Final Stop, B-Cell Vaccine. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by GenScript. Studying neutralizing antibodies is of critical importance in hepatitis C virus research, since no vaccine is available. Hepatitis C-related diseases still cause 400,000 deaths a year worldwide. In this webcast, we will hear about an innovative approach to reconstruct the virus neutralization space from Dr. Dorothea Bankwitz of the Twin Core Center of Experimental and Clinical Infection Research in Hanover, Germany. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today.
Is on.
Now, over to Dr. Bankwitz.
Today, I would like to guide you to hepatitis C virus neutralization space. Our final stop is a HCV B-cell vaccine. Even so, we will not reach the final stop today. We will be on the right path, and you will learn what to consider when designing a B-cell vaccine for highly variable viruses. Hepatitis C virus is still a major global health burden, with 71 million people infected worldwide. Per year, there are 1.75 million new infections, and the leading cause of new infections are unsafe healthcare procedures and injecting drug use. HCV infection causes 400,000 deaths per year, and it is a common cause of liver transplantation. We still have these high numbers. Even so, there are very efficient direct-acting antivirals with cure rates of 95 to 99%. So what is the problem with treatment? The problem is that by far not all patients are diagnosed, and those who are diagnosed, not all have access to treatment. Furthermore, treatment cannot prevent reinfection, and therefore an HCV vaccine is still highly needed. So what is the current situation in HCV vaccine development? Here's a very impressive comparison of HCV vaccines that are in clinical trials and SARS-CoV-2 vaccines in clinical trials. For HCV, so far, only 3 vaccines entered clinical phases: 2 protein subunit vaccines and 1 viral vector vaccine. One of the protein subunit vaccines is composed of core protein, designed to induce T-cell immunity. However, T-cell response was only detected in 25% of the participants, and therefore this vaccine did not enter clinical phase 2. The other protein subunit vaccine candidate is composed of recombinant E1, E2 envelope glycoproteins. This vaccine is designed to induce B-cell immunity. However, cross-neutralizing antibody responses were only elicited in a few subjects, and therefore this candidate was not advanced further to high-risk group. The third vaccine candidate is an adenovirus-based vector that expresses HCV non-structural proteins and that is designed to induce T-cell immunity. Earlier this year, it was reported that this HCV viral vector vaccine unfortunately failed because it could not induce protective immunity in a high-risk group. So, all 3 vaccines failed to induce significant protective immunity, and that is the situation we have more than 30 years after discovery of HCV. So in contrast, 1.5 years after first detection of SARS-CoV-2, we have 4 approved vaccines and more than 100 vaccines in clinical phase 1, 2, or 3. Of course, we have to keep in mind that we have 2 completely different vaccine targets here. So let's look in more detail what makes HCV such a special target. So in our study, we focus on development of a B-cell vaccine and therefore are interested in antibody responses targeting the HCV surface glycoproteins E1 and E2 that are embedded in the lipid membrane of this positive sense single-stranded RNA virus. So with respect to vaccine development, E1 and E2 have 2 important features. So on the one hand, these 2 proteins have highly variable sequences, and on the other hand, a virus with a given sequence shows remarkable structural flexibility. First, a few more words on sequence variability. Here's a genetic diversity of 3 envelope glycoproteins from 3 different viruses are illustrated, and the scale bar shows a 5% amino acid differences. The diversity of HCV E1E2 is in the same range as diversity of influenza hemagglutinin and HIV-ENV. So the variability of these immunodominant surface proteins is one of the reasons that an effective and universal vaccine against these 3 viruses has so far not been really successful. But for HCV, the sequence variability is only one part of the challenge in B-cell vaccine development. The second challenge is to understand and to consider the structural flexibility of E1 and E2. So for E1, unfortunately, there's no structure available. Therefore, only E2 is illustrated here. It was a major challenge also to solve the structure of V2, and still the structure that is solved is incomplete. So the hypervariable region 1 is missing, and most likely this region is also important for the structural flexibility of the whole protein. So a virus with a given E2 sequence can adapt different conformations. And if we focus on the epitope 1, you can see that epitope 1 can adapt a beta hairpin conformation, a semi-open conformation, and an open conformation. And patients have antibodies binding to all these different conformations. So also for other epitopes, different conformations are described. And this structural flexibility of E2 is still incompletely understood. So, obviously, this extreme diversity of HCV is a major obstacle for vaccine development. However, spontaneously clearance of HCV happens in about 25 to 30% of the patients, suggesting that chronic HCV infection is indeed preventable. And HCV clearance is also closely associated with the presence of strong and early neutralizing antibody responses that target HCV E1E2. So we in our study wanted to establish a tool that facilitates characterization of neutralizing antibodies induced either by natural infection or by vaccine. And in addition, we aimed to develop a tool that can be used for prioritization of vaccine candidates. So that's where our tour to HCV neutralization space starts. As I mentioned, a high number of genetically and functionally diverse HCV variants is existing. We now aim to group these variants according to their neutralization, and therefore we make use of patient antibodies that reflects the majority of existing antibody responses. Next, I would like to show you the set of viruses that we selected to start our study with. We selected 13 viruses. These viruses differ in their E1, E2 sequence. And we used full-length viruses derived from cell culture. These viruses have a vanilla luciferase reporter. And shown here is a phylogenetic tree based on E1, E2 amino acid sequence. So HCV has 8 major genotypes that differ up to 30%, already indicating the really high variability of HCV that is really remarkable. And all the viruses that we selected are indicated here as circles. So our viruses represent 5 different HCV genotypes and 9 subtypes. So therefore, our panel provides a broad overview of HCV diversity. And here I have to mention that it is very difficult to create cell culture-derived HCV with diverse E1, E2 sequences that, on the one hand, efficiently produce infectious virus, and on the other hand, do not need cell culture adaptive mutations, which could potentially influence neutralization properties. However, we succeeded in creating new virus variants, and now we have 13 viruses without cell culture adaptive mutations in E1 and E2. So, intuitively, one would assume that taking one isolate from each group would probably reflect all circulating HCV strains. That is also what was done in numerous studies. But is that really the best idea? That's what we want to address in our study. And of note, we also included 7 genotype 2 derived envelope proteins to test the hypothesis if viruses belonging to the same genotype have similar neutralization profiles. So next, we wanted to explore the diversity of our panel at the structural level. And therefore, we mapped the sequence divergence between the 13 viruses onto the crystal structure of the E2 ectodomain. And both conserved and non-conserved residues are over the surface of the entire glycoprotein. Next, we profiled the reporter virus panel with the neutralization assay using well-characterized monoclonal antibodies binding to well-characterized diverse epitopes. One out of these 7 monoclonal antibodies is shown here. HC8048.26 is binding to epitope 2, shown here. And what you see is that this monoclonal antibody can neutralize some viruses very efficiently, other viruses almost not at all. And this indicates that our virus panel is functionally diverse with respect to neutralization. We next determined the antigenic relationships between these viruses, and to do so, we decided to use polyclonal patient IgGs instead of monoclonal antibodies because polyclonal patient IgGs are reflecting the diverse antibody response during natural infections. So we could do so by using patient cohort of 100 patients infected with HCV genotype 1 to 6, similar numbers of genotype 1 to 4 infected patients, and only a few samples originated from genotype 5 or 6. So we purified antibodies to avoid bystander effects by serum components. And we performed neutralization assay using our 13 virus panel and the polyclonal patient IgGs. Our readout is based on the vanilla luciferase reporter virus, and the first data set that we obtained is shown here. So this data set includes 1,300 virus patient IgG neutralization pairs. And what we observed is, on the one hand, a vast difference in neutralization between the different patient IgGs. So almost all patients have antibodies that can neutralize at least one virus. Here's the genotype 5a virus. And on the other hand, we have observed vast differences in virus susceptibility to neutralization. So rank ordering all these viruses according to neutralization sensitivity showed that the panel has a wide and evenly distributed spectrum of neutralization susceptibility, with genotype 2a, J6, being most resistant, and genotype 5a, the isolate SA13, is the most sensitive virus. And next, we aimed to categorize the HCVcc panel into virus groups with similar neutralization phenotypes. Therefore, we used these data here and collaborated with bioinformaticians, and they performed metric multidimensional scaling. This metric multidimensional scaling is shown here, and what is done is that neutralization data of virus patient IgG pairs are projected into a 2D neutralization map with one square in the map corresponding to 10% residual infectivity. And the distance between each patient IgG and each virus reflects the neutralization efficiency, with small distances representing very efficient neutralization and large distances reflecting an inefficient neutralization. So let's take an example here. The genotype 2a virus J6 can be neutralized very inefficiently by almost all patient IgGs. So for simplicity, we will now remove the antibodies from the map, and now only the 13 viruses of our screening panels are left. Next, we used k-means clustering to identify that the HCVcc strains segregate into 6 distinct neutralization clusters, and viruses in one given cluster, like for example here, this cluster, share similar antigenic properties. All viruses in here have common features with respect to neutralization. Next, we were interested how clustering based on neutralization profiles correlate with phylogenetic relationship of the viruses. Shown here is a sequence-based clustering versus a neutralization-based clustering. So viruses belonging to the same neutralization clusters are indicated by colored bubbles here in the phylogenetic tree. And some clusters consists of viruses of 3 different genotypes. For example, here, this cluster consists of viruses that belong to 3 different genotypes. And also very important is the observation that the 7 genotype 2 isolates belong to 4 distinct neutralization clusters. Therefore, viruses belonging to the same genotype do not have similar neutralization behavior. So we next examined if a subset of viruses can also reconstruct the neutralization space of the entire panel. So in more detail, we selected reference virus for each neutralization cluster. So we scaled down from 13 viruses to 6 viruses, and we wanted to evaluate if these 6 reference viruses can reflect neutralization space that we obtained by the 13 virus panel. Therefore, we calculated a cross-neutralization index, shown here, that takes into account both the antibody breadth and potency. And we used this cross-neutralization index to rank order the patient IgGs from the 6 virus panel and the 13 virus panel. And we did a Spearman correlation, and we found a very good correlation. And therefore, reducing the number of strains did not compromise the precision of scoring both breadth and potency of the neutralizing antibodies. And actually, we believe that the cross-neutralization index for the 6 virus panel is even more accurate, because calculating this index using several variants belonging to the same neutralization cluster results in an unbalanced cross-neutralization index. So the 13 virus panel, as I have shown you, has several viruses for some of the neutralization clusters, but not for others. So our index would be biased for these clusters having more viruses. And therefore, we believe that our 6 panel is even superior. Our next question was if we can reconstruct the neutralization also by doing a more simple ELISA-based antibody binding assay. And the regression analysis of neutralization and binding shows that we could not observe a correlation, and therefore we conclude that we cannot use HCV E1, E2 ELISA binding assay to reconstruct neutralization space, and that the 6 virus panel is the best we have in hand to perform studies to characterize antibody responses after vaccination and during natural infection. Therefore, in the next step, we wanted to further validate the performance of our 6 virus panel also using larger patient cohorts. So why studying larger patient cohorts for antibody specific responses? To study natural immunity in patients seems to be a prerequisite in HCV vaccine development, to be informed how to design an antigen that can be used as a vaccine candidate. And that is why we want to use our antigenically balanced virus panel to study bigger patient cohort. So we had access to almost 400 patients chronically infected with HCV genotype 1 to 5, and after purifying the polyclonal patient IgGs, we again performed neutralization assays with our 6 identified reference viruses. So the results are shown here. In this heat map here, we rank ordered the patient IgGs based on their cross-neutralization index and selected the 2% with the highest cross-neutralization index as elite neutralizers. And to prove that our screening with the 6 virus panel indeed identified patients with antibodies that are cross-neutralizing against the full spectrum of the 13 virus panel, we then used the 13 panel to perform neutralization, and indeed all elite neutralizers had cross-neutralizing antibodies neutralizing all viruses at least 50%. This is a strong indication that our screening approach is reliable and can be used to study natural immunity as well as vaccine-induced immunity. Finally, we wanted to use the entire neutralization data from the first patient cohort with 104 patients, together with this patient cohort here, this 392 patients, to further analyze our Zika virus screening panel regarding the performance and how balanced the virus panel is. So again, we projected the neutralization data of the patient IgG virus pairs into a 2D map. And what we observed is a dense cloud of IgGs from patients infected with different genotypes. Genotypes are actually well intermixing with each other, suggesting absence of genotype-dependent subclustering. That means that, for example, antibody profiles in genotype 1 infected patients do not differ from antibody profiles in a genotype 3 infected patient. So there is a very similar diversity of antibody responses in patients infected with different genotypes. And this has important implications for vaccine design, since the genotype of the antigen does not seem to predict a specific antibody response, but other features of the antigen, as for example, specific structural features, are important to consider. So, what are the important findings from our study and what are the implications for vaccine design? So, we performed high resolution profiling of about 500 patient derived IgGs to develop, to calibrate, and also to validate our novel screening method that can be used to quantify HCV antibody responses. Furthermore, HCV viruses could be mapped to 6 neutralization clusters, in part composed of viruses from different genotypes and subtypes, indicating that neutralization clusters differ from sequence-based clustering. Moreover, we could show that multidimensional scaling can be used for deep phenotyping of antibody responses. So, what are the implications from our findings for future HCV vaccine development? So, our newly established set of reference viruses will facilitate to characterize antibody responses in natural infections and will help to predict antibody responses induced by vaccines. So we believe that our in vitro tool is highly relevant because there is no immunocompetent small animal model for HCV, and reliable in vitro tools are therefore indispensable. Finally, we can conclude from our findings that vaccine candidates addressing viral functional diversity and not genetic diversity may induce superior protective immunity. With this, I would like to stop. Finally, I would like to thank a number of people because this project was a real team effort. First of all, I would like to thank all members of our institute, especially those who contributed to this work. In addition, I would like to thank Alice McHardy and Akash Bahai, with whom we closely collaborate. They are the bioinformaticians. And I would also like to thank our clinical partners. Funding was received by the German Center of Infection Research, as well as from the Excellence Cluster Resist. And here I would like to end, and I would also like to thank you for your attention, and I'm happy to take questions.
Thank you very much, Dr. Bankwitz, for that presentation. It is now time for the question and answer session. To ask a question, please type it in where it says type your questions here, and then press submit. So, Dorothea, to get us started today, I wanted to ask you, you identified a number of patients with potent cross-neutralizing antibodies. Why were these patients not able to clear the infection?
Yeah, we believe that a very early and efficient antibody response is needed, and those patients that already proceeded to chronicity have broad antibody response, but too late. So the infection is already established, and it's basically the timing of the antibody that is also important, and therefore we see cross-neutralizing antibodies in these patients, but these antibodies seem not to protect from infection.
Thank you very much. It's crucially in that case. Moving on, I wanted to ask you, how do the in vitro neutralization assays reflect the in vivo situation?
Yeah, so we use cell culture derived HCV, and in addition we used a hepatoma cell line. So basically the cell culture HCV seemed to reflect the in vivo circulating virus quite good. So it's also associated with lipoproteins, and the entry process seems to be very similar. So we believe that there is a good correlation. Of course, in vivo is still different, and using primary hepatocytes in addition to hematoma cell line would be also a next step, but is very difficult to really do in the lab. But we believe that our in vitro approach indeed reflects in vivo situation.
Okay, thank you very much for that clarification. The next question asks about pseudoparticles. HCV pseudoparticles are easier, to handle. Is it possible to clone the identified E1, E2 sequences into HCV pseudoparticles and to get comparable neutralization results?
Yeah, so indeed these retroviral HCV pseudoparticles are used in a number of studies. Most panels that are used are also genotype 1 biased. And the problem with HCV pseudoparticles is that these pseudoparticles are not associated with lipoproteins. Therefore, they do not really reflect the in vivo situation. And it seems that the pseudoparticles are more susceptible to antibodies than the full-length HCV cell culture derived virus that we use. And therefore, considering these, we think that using the HCV full-length virus are superior. Even so, it's possible to clone our E1, E2 sequences into pseudoparticles, but we think that's really an advantage of our studies, that we now established a tool with HCV cell culture derived viruses and patient antibodies that really reflect better the situation in vivo.
Okay, thank you. Our next question asks about genetic diversity in GT2, E1, E2. What is the level of genetic diversity in the GT2, E1, E2 sequences of chimeric viruses, particularly whether the question is asking whether or not I think there's any span in known neutralizing epitopes.
So the genetic diversity in one genotype is around 15%. And so that's basically also what our 7 genotypes of 7 isolates of genotype 2 are also showing with respect to diversity. And the second question to these genotype I missed. Can you repeat it?
If there's any known span of neutralizing epitopes.
Yeah, I mean, there are epitopes identified, a number of epitopes that the antibodies can bind to, indeed. And so these are already well characterized, but it's not known if all epitopes are already identified.
Okay, thank you very much. Is it possible to further reduce the number of viruses to perform a larger screening approach that's more efficient, or to save antibodies?
Yeah, indeed, that's possible. So we use this 6 virus panel, but to identify, for example, elite neutralizers, it's also possible to do a pre-screening with one isolate, and we identified the genotype 2R isolate to be best here. So that correlates quite good to the 6 panel with respect to cross-neutralization index, and can be used to save antibodies and time to do screening approaches.
Okay. A quick reminder to our live audience, you still have time to ask a question. To do so, please type it in where it says type your questions here, and then press submit. So with you, we learned that it's not the global sequence of E1, E2 that predicts neutralizing features. Is it possible to identify a smaller sequence pattern of E1, E2 that predicts the affinity to a specific cluster?
Yeah, so indeed, we are now aiming to enhance the number of test viruses that we can then analyze in the neutralization map. And that would be a prerequisite to identify these sequence or structural patterns that are important to group the virus to specific clusters. So, but therefore, we are still in the process of enhancing the number of viruses that we can use. And we hope to indeed finally find sequence or structural features that allow us to predict from the sequence where this virus clusters to.
Thank you very much. A further reminder to our live audience, you still have time to ask a question. To do so, please type your question in where it says type your questions here, and then press submit. Our HCV immunity seems not to be directly coupled with a certain genotype. I'm in clarifying conserved epitopes of the 2% broad neutralizing antibodies, for example, in mass spectrometry. Would this narrow down the ideal vaccine candidate? If so, what do you think?
Yeah, so using these elite neutralizers that we identified, we indeed go on to further analyze the antibody responses, doing, for example, B-cell cloning, to learn more about the epitopes these antibodies are binding to finally use the identified epitopes in a vaccine candidate.
Okay. Thank you very much. And that is where we will finish for today. I would like to thank Dr. Dorothea Bunkwitz for her presentation and for answering your questions today. I would also like to thank the sponsor, GenScript, and you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at www.nature.com/webcasts. Thanks for watching, and I hope you'll join us again soon.